Announcements, that's the usual thing at this time of day. Just to say that Sparkle engineers and our OHBM volunteers have been working very, very hard overnight and during the day, trying to deal with that pesky little problem in the poster hall. And happily, that has been solved. And I have to make a big shout out to our own Soichi Hayashi for doing that. Thank you, Soichi. We really appreciate it. So, in that vein, be sure to visit the poster hall today for our evening poster session. And we've actually invited all poster presenters to come on out and stand by their posters, irrespective of what category they are in. So that would be a nice opportunity for more interaction. In our second time block, we have OHBM Club Night, our virtual club night, and Club OHBM, which can be accessed from the landing page on Planet Brain from the bottom left icon of your screen. And you can do that during the social hour. I'm going to also congratulate the OHBM fellows, and then hopefully we can have a nice slide of them. There's 3 of them, and here they are. And these are familiar faces to people who, these 3 individuals, the 3 amigos as I call them, have done a tremendous amount of work, hard work for OHBM over the years. And so we celebrate them and their effort today. Now, I have the pleasure of announcing, of course, the Glass Brain Award again. And I want to just reiterate that the Glass Brain Award went to Dr. Peter T. Fox. And before we show his acceptance speech, let me give you a quick bio of him. And I mean, I have to be quick, but he's such an accomplished scientist. He did his education on the East Coast, BA in Liberal Arts at St. John's College in Annapolis in Maryland, followed it up with an MD at Georgetown University School of Medicine in Washington, D.C., and then did 2 internships in medicine. The first one was in medicine at Duke University Medical Center, and the second one was in neurology at Washington University Medical Center. He has also, as a clinician, formal research training, and he started out as on an NIH T32 grant, which was awarded, a training award, which was awarded to Washington University Department of Neurology and Neurosurgery. And his project there was to develop and apply novel methods for mapping sensory motor brain function. We're using O15, positron emission tomography. He then followed that up with an individual K award, a clinician scientist K07 training award from NINDS. And the goal there was to move those new PET methods to study higher order brain functions, such as language and attention. And of course, the rest is history there. In terms of positions, he has moved from the Johns Hopkins University School of Medicine, where he was a founding member of the Mind-Brain Institute. And ever since 1991, he has been at the University of Texas Health Sciences Center in San Antonio. And he was the founding director there of the Research Imaging Institute. And that's an organized research unit dedicated to development and application of advanced imaging methods. And he's there to this day as its director. He's also the vice chair of research and research education in radiology. There he holds the Malcolm Jones Chair of Radiology. He's also professor in the departments of neurology, psychiatry, and physiology at UT Health San Antonio. And he is the member of quite a number of graduate faculty programs at UT. He served overall and trained 31 predoctoral trainees, 13 postdoctoral fellows, and 17 faculty under these US federal training awards. Amazing, amazing track record. And last but not least, he was the founding editor of the journal Human Brain Mapping, and he founded that in 1992. It's gone from strength to strength to the point where now in 2021, he's assisted by 3 deputy editors-in-chief, Jack Lancaster, Gary Egan, and Simon Akoff. So let's roll Peter T. Fox's acceptance speech for the Glass Brain Award.
Thank you for what I assume was an embarrassingly generous introduction. As you just learned, my name is Peter Fox. I've been involved in the Organization for Human Brain Mapping since before we knew what we would call it. For me, it's been a labor of love, contributing to OHBM and participating in all 27 annual meetings. This is the 8th year that OHBM has presented the Glass Brain Award. Reviewing the list of prior awardees is both humbling and heartwarming. Marcus Rakel was my first mentor. His steadfast support in the face of controversies I stumbled into was really foundational for my career. Alan Evans, David Van Essen, Carl Friston, and Susan Buchheimer, all Glass Brain recipients, have been collaborators, co-authors, and good friends for decades. Leslie Ungerleider and Carl Zillis are no longer with us. Peace be with them. But both contributed enormously to this community and will be long remembered. It's an honor to be in their company once again as a Glass Brain recipient. For those of you who are new to this community, it's a remarkable collective undertaking. Members here come from a host of disciplines, including cognitive and clinical psychology, neurology, neurosurgery and psychiatry, physics and engineering, statistics and computational biology. Despite this diversity, we're brought together by a commitment to systems neuroscience, very broadly considered. Simply put, we all share a passion for understanding the human condition, and we see neuroimaging as providing the best tools for that purpose. It's a remarkable group of like-minded but amazingly diverse individuals. I'm grateful to OHBM leadership for recognizing my contribution to our common goals. I thank all my collaborators and colleagues from over the years, particularly my former and present trainees. I want to recognize my family, and in particular my wife, Sarah Beth, for accommodating the long and often irregular hours and the frequent travel of our scientific lifestyle. To close, I'd like to thank everyone in the audience for participating in our second and hopefully our last virtual OHBM. I look forward to seeing you all in person in Glasgow for OHBM 2022. Thank you. Have a great meeting.
Hello, I am Michel Thiebaud de Choten, and I'm here to introduce our keynote speaker, Anna Wang Roe. For those of you who don't know, Dr. Anna Wang Roe is the director of the Interdisciplinary Institute of Neuroscience and Technology at Zhejiang University. She pioneered work in the rewiring of the neonatal ferret brain. And if, like me, you're asking yourself whether cortical functional specialization or connection are coming first in the brain, here is one of her papers I tell my collaborator and students all the time. She rewired the primary visual cortex onto the auditory cortex in the neonatal ferret and observed a reorganization of the receptive fields in the auditory cortex that took on visual functions, really. And it seems connections come first before functional specialization. Her career is paved with exceptional neuroscientific contribution like this. And this is going from anatomy to question about the visual system and the veracity of perception itself. Most recently, I worked on toward building prosthetic for the visual system. She has published over 120 peer-reviewed articles, and she organized numerous international conferences and symposia and actively participates in review panels and editorships. In recognition for her contributions, she has received Sloan Whitehall Packard Fellowships and has been elected member of the International Neuropsychological Symposium, as well as SPI Fellow and AAS Fellow. That is why we are very lucky to have her giving us a live lecture, well, a recorded lecture today entitled INS-fMRI, a method for mapping mesoscale connectomes in the non-human primate. Thank you.
Thank you so much for such a nice, nice introduction. I really appreciate that. So let me get started here, and I will just share my screen. Okay. So I trust everyone can see this. Okay. So my talk today is going to be about a mesoscale-connectome. It is basically about how to make a Google map of networks in the brain at columnar scale. This paper was chosen as top 10 medical technologies in China in 2019. So this is what this little cartoon represents. So I will just get started by very generally saying that we study how the brain networks underlie behavior, communication, vision, emotion, social behavior. These are all behaviors we're very familiar with. I study these behaviors by studying non-human primates. Non-human primates are quite similar to humans in terms of their behavior, the way they use their eyes for vision, hand use. They share lots of cognitive and memory functions that are similar to us, as well as limbic and social behavior. So they're really quite good models for studying human behavior as well as human brains. And I'm going to tell you one reason why they're good models is because monkey brains are similar to human brains in the sense that they are organized in units commonly referred to as columns or patches or domains. These are repeating units in the brain, and they have different functions. Different types of columns have different functions, and I've spent my career studying them. They are organized as shown previously by Hubel and Wiesel, for which they got the Nobel Prize in 1981, that there are very systematically laid out information columns in visual cortex. And these have gone on to be shown elsewhere in the brain as well. Pashko Rakesh and his colleagues have done much to show that these columns are genetically sort of instructed. They're protomaps for columnar organization very early in development. And so this structure is very much a primate-specific structure. These columns are quite small. They're less than 0.5 millimeter, a couple 100, 300 microns in size. And so here I just show a little summary of some of the maps we've seen in visual cortex using intrinsic signal optical imaging. Here you see that there's a monkey brain on the bottom left, and there's a window implanted on the brain such that we can see activity on the cortex through this window. Based on hemodynamic signals, we can see that different maps emerge as we show the monkey different types of visual stimuli. So we now know that, for example, this ocular dominance column map in blue encodes eye-specific signals that are important for depth perception. Orientation maps in pink are important for shape perception. Color maps are important for color perception, and motion direction maps are important for motion perception. Each of these maps is comprised of very small columns, and they are encircled in the little red circles inside each map, and they're 200 to 500 microns in size. Now, if you were to multiplex these 4 maps into one, you would see something akin to what's shown on the bottom here. And one can start to see that you can decode different objects by looking at these maps. And I can tell you that I can tell what the monkey's looking at without looking at the monitor. I know what he's looking at just by seeing these maps. It's very cool. Okay, so these maps also exist in other areas of the brain, not just visual cortex. There's somatosensory cortex, primary motor cortex, even prefrontal areas, and they underlie different kinds of behaviors. So columns are pretty ubiquitous throughout the brain. It's not been proven, but given what I've seen so far, I do believe that if we find the right parameters to probe each area, we would see columnar organization. And much of the anatomy that has been done also predicts this. So in the last decade or so, there's been a lot of studies on connectomes. Connectomes is a study of all connections in the brain. And these have used functional approaches as well as structural approaches. And all of these connectome projects in Europe, US, Japan, China, they have produced maps like that on the right, where you have maps of different connectivity patterns in the brain. Some of these are macro scale and some of these are micro scale, that is at cellular level. The micro scale maps are anatomical, and in the macro scale maps typically used in human studies are based on resting state and have been hugely influential in our understanding of brain function. Now, the typical resolution for obtaining these maps, at least in humans, are 2 to 3 millimeter voxels. So this is what I am addressing today, which is that this resolution is not sufficient for looking at the basic units, which are 0.5 millimeter or less. And so that's why I just want to make the point here that if you had a voxel on the right here, represented by the white square, and within this white square, there were 2 different kinds of domains, such as color domain in thin stripe and disparity domain in thick stripe. Well, we would see that from resting state that this one voxel connects to both dorsal and ventral pathways, where in fact, only the thin stripes project to the ventral pathway and only the thick stripes project to the dorsal pathway. And so low spatial resolution can lead to misleading understandings about brain connectivity. So I'm going to tell you today about a mesoscale map. I just want to let you know that this is why it's important to look at the mesoscale level, the submillimeter level. And so it's been challenging because of the spatial resolution that is required. So I'm going to tell you a little bit about brain architecture. We've already talked about columns, the functional units in the brain, but we also know from anatomy that these functional units are connected to each other in very specific ways. So this is a picture of an anatomical injection of tracer in the middle circle, the darkest one, and what's labeled are label of different columns nearby. And all of these columns share the same orientation and selectivity. So this anatomical picture represents a functional network in the brain, representing an orientation selective network. So this has been known that columns are connected to other columns, at least at a very local region. But what's unknown is what are the organization of these connections as you move from one column to the next? How do the connected patterns shift or change with a changing central node? Do these networks, these columnar networks, exist at a brain-wide scale, or is it only at a very local level? Furthermore, we'd like to know whether we could do this in vivo without sacrificing the animal. So anatomical studies do require sacrifice of the animal and reconstruction of all the tissue, and it's quite time consuming. And finally, we'd like to know what is the relationship of these networks to behavior. So I'm just gonna motivate this by saying that, well, we know that these connections are there. Is it possible that if we stimulate a central node, we could activate the connected nodes and thereby see the entire network? Well, so I've been spending some time trying to develop ways to study columnar connections in vivo. And here are 3 of the ways that we've tried to do this using electrical, optogenetic, and infrared neural stimulation methods. Each of these can be used to stimulate single columns, however, with different degrees of difficulty. So electrical stimulation has been around for, a century or more. It is a little more difficult to confine the electrical effect of the electrical stimulation to a single column because there's current spread, and that makes it a little more challenging. But as you can see on the picture on the left, if you try hard enough, you can almost do it. And you can activate some columns nearby, indicated by the red dots. Using optogenetic approaches in non-human primates can be done, and we have done it. Lots of people have done it. It's a little more time consuming and challenging because you have to inject viral vectors to transfect the neurons so that they express light sensitive channels, such as channel rhodopsin. Well, this can be done, but it is not so convenient and not so reliable in non-human primates. And furthermore, you can only stimulate in the locations where the optogenetics has taken. So we then turn to another method called infrared neural stimulation, or INS. And this method uses a long wavelength light, 1.875 microns long, so that it's not visible to your eye, but it's quite effective, and we pulse it at in very brief pulses of 0.25 milliseconds wide. So it's really brief, like a brief like that, and then we pulse it for half a second, so it's like a very small little stimulus. And when this is delivered at 0.1 to 1 joules per centimeter squared via a 200 micron fiber optic, it achieves neural activation. And so we've used this connectivity patterns in the brain at local scale in visual cortex and somatosensory cortex, looking both at lateral connections as well as feed forward and feedback connections. So it is feasible to map such columnar networks in vivo at local scale. We next wanted to see, well, can we do this at global scale and look at brain-wide scale networks? Well, so here we have shown that, this slide is a little out of place, but we have shown the INS can induce activation of neural activity in the upper left here, as well as it can induce inhibit, deliver it with the right parameters. So it is also intensity dependent. And on the right, on the top, you can see that with increasing intensity from 0.1 joules to over 1 joule per centimeter square, you can get an increasing response, neural response, as revealed by optical imaging. And you can see that the light profile of the light delivered with a 200 micron fiber is very focal. It's not much more than 200 microns in width and about 300 or so microns in depth. So it can go into the tissue a few hundred microns distant. We've also shown that when applied to visual cortex, that can activate selective columnar networks in visual cortex. Methods can't. And furthermore, this method can be used in a way that is non-damaging histology from these cortices appear normal and not damaged, at least within these parameters. And you can repeatedly use this method on the cortex without damaging the tissue. You have normal optical and electrical responses, normal behavior, and good health of the animal. So when we wanted to examine the possibility that this method could be used at brain-wide scale, we then went to MRI, and in fact to ultrafield MRI. The 7T-MRI in our center is a Siemens magneton machine. And here you see images from a cat brain where we stimulated cat visual cortex in area 17. You'll see the red star asterisk represents the stimulation site. You can see as the intensity increases from 0.1 up to 1 joules per centimeter square, you can see an intensity increasing bold response. And you can see that the bold response follows the stimulus quite well. And the intensity relationship is shown in the bottom right. So the greater the intensity, the bigger the bold response. So we can measure that. Now, so the idea is that we wanted to stimulate a single column in the brain or a single site and see differential networks. So the yellow activated network would be stimulated from one node, and the red activated network stimulated from a second node. And this is quite different from previous work where you have to do anatomy, which requires tracer transport time, animal sacrifice, and time-consuming reconstruction. Optogenetics, as I mentioned, requires viral transfection, and then electrical stimulation suffers from current spread and is also a little less easy to use in the MRI. So when we combined, the INS with 7T-MRI, we found that we could develop this in vivo method that could be used rapidly. You could see the networks right online at the console. It's mesoscale. One can induce activation of small nodes and their connected sites. Does not require animal sacrifice, and there's no time-consuming reconstruction. Okay, and furthermore, you can do this repeatedly in the same brain, time after time, and we have done that. So let me preface what I'm going to tell you next by saying that there was a lot of groundwork to be done before we could achieve this mesoscale type of mapping. So in our team, we have MR physicists who deal with hardware, designing custom array RF coils, custom pulse sequences, optical people with optical expertise to design fiber bundles, as well as different types of stimulation parameters. People who dealt with animals, surgical methods, as well as animal behavior, training monkeys in the MRI, achieving high spatial resolution structural, as well as high spatial resolution functional mapping. And finally, engineering of other methodologies for 7T use. So I'm going to tell you about two studies that were published. One, both of them show that this method can work at brain-wide scale. And the first study is on visual system, and the other is on the limbic system. So I'll tell you about the vision vision study first. This study was done well, both in cats and in monkeys. And this study, I'm going to just tell you very briefly that we stimulated in area 17, and you can see that we chose the cat visual system because the anatomy is very well known. Stimulation of area 1718 achieves activation in area 18, 19, AMLS and PMLS, as well as higher order areas such as anterior ectosylvian and cingulate visual area. And so we predicted that these areas would be activated by single site stimulation. And again, we chose a 200 micron fiber because we wanted to confine the stimulation to a single column. And here are all the slices in one cat brain scanned in the 7T-MRI with the stimulation at the red asterisk. And you can see this was at the 17-18 border. We see activations in 18 and 19. We see activation in LGN and contralateral visual areas, AMLS, PMLS, as well as anterior ectosylvian area, as well as cingulate visual area. Furthermore, we showed that this stimulation was intensity dependent, not only at the stimulation site, but at the connected sites as well. So this shows that we are able to study mesoscale brain connections in vivo using this approach. So we were pretty excited by this. I mean, that we could get what we predicted. So we wanted to try something different, which was to see if we could so we chose as a test bed, we chose the amygdala in the macaque monkey. In particular, we chose the basal nucleus of the amygdala. The amygdala is a reasonably well-studied region of the brain. The connections are well known. So David Amaral and his colleagues, there are numerous investigators who have shown the anatomical connections from different subnuclei within the amygdala. A couple of the strongest connections are with the cingulate and with the insula, and I'll just show you an example of the cingulate. The other study, I'm going to show you this stimulation using a 200 micron brain into the basal nucleus of the amygdala. And the amygdala is a really central nucleus in the limbic system. It essentially is an interpreter of how the outside environment, the sensory inputs that come in, are understood by your cognitive areas and how you should respond to those stimuli in a contextual manner. So there are lots of connections with sensory areas, with prefrontal areas, with motor areas. Those are quite integrative region in the brain. So this simply shows you some slices through the amygdala, and you can see that in C, the fiber has been inserted into the lateral part of the amygdala, and it's being positioned into this region. And you can see in D through F that as the fiber reaches these amygdala regions, if the fiber is in that slice, you'll see a black dot where. And as you go from slice in D to the next slice in E to the next slice in F, that dot just disappears in F. And so we know that the tip of the fiber has reached E, but not F. So we can identify the location of the fiber tip to within 0.3 millimeter resolution. So this is something that is reasonably reliable for trying to understand the sub-organization of single subnuclei within the amygdala. And we also showed that stimulation within these subnuclear regions is intensity dependent. Okay, just a simple showing you that we can see anatomy online. It's quite direct. And here's a penetration with the fiber optic. where we stimulated 3 different sites, 2 in the amygdala and 1 in the putamen here. So we can identify where these sites are, and with the expert eyes of Kathleen Boucard and Liz Romansky, we were really a little more confident about where these sites were. And so we can stimulate, position different fibers in different locations, and stimulate multiple sites within a single amygdala. And so this is just an example from one site. You see that the stimulation at the tip here is quite visible, and the slices throughout the brain, you can see that these are almost all the slices throughout this monkey's brain. There are not that many voxels. In fact, it seemed relatively sparse and quite specific. The activation seemed to be in very specific patterns and very specific sites. And actually, throughout the whole brain of the macaque monkey, which has about 30,000 voxels, we typically would see voxels activated depending on the thresholds that you used. And so here is just shows you 3 examples from 3 hemispheres of 2 monkeys with the fiber optic penetrating into the basal amygdala. And each of these 3 reveal activation in the cingulate cortex, which is areas 23 and 24. And you can see that these activations appear very focal and patchy, spaced along the anterior-posterior extent of the cingulate cortex. This is true in case 1 in D, case 2 in E, as well as in case 3 in F. As well, you see there are a couple activations in the medial prefrontal cortex, areas 25 and 32, as well as occasional activations in retrosplenial cortex. And so this is quite consistent with the known anatomy of basal amygdala with cingulate. However, what is new or more new is that these connections are quite focal and patchy in nature. And I should also add that this activation pattern in cingulate cortex maps well onto the known limbic motor regions of cingulate cortex. That is, they appear to map well with the face and hand regions of cingulate. And it's possible that stimulation at this basal nucleus site in the amygdala activates a circuit that is related to facial and hand gestures during limbic response. So given this result, we were encouraged and we decided to try a connectome experiment in which we would activate or stimulate many sites within a single amygdala. So here are just a bunch of slices. I'm going to show you a bunch of slices through a single amygdala. And you'll see that there are different penetrations and different sites. And finally, over Chinese New Year this year, my students and postdoc, Xu Guohua, did a lot of work and generated this matrix where we had 55 stimulation sites and all the connected brain sites from those stimulation sites. And this is that matrix. We're still working with it. We've written up some of it and are just about to submit it now, but it's very exciting. So all the connections of one amygdala are represented in this picture. On the bottom are all the brain areas, and in the right-hand column are all the stimulation sites. We had multiple runs per stimulation site. So it's about under 200 runs. And this is collected over 4 days and involved students from multiple labs. So this allows us to compare networks within an individual. So this is something that is not easy to do with anatomy, especially at this resolution where we can move the fiber millimeter by millimeter and understand the network connected to each of those sites. And so this also allowed us to study the topography of the connections, how the networks shift as the stimulation site shifts. Okay, so just to summarize, INS-fMRI is a method for mapping mesoscale circuits in vivo. We combined infrared neural stimulation with a fine fiber optic to activate single sites within animals imaged in a 7T MRI. And our stimulations showed that indeed we could get specific activation patterns that were predicted in anatomy and furthermore revealed new aspects of connectivity. This method is in vivo. It does not require viruses, reveals mesoscale connections, and furthermore, we can know the functional identity of the stimulated site and or the activated site. It's fast. That means you can see it appear right on your console as you're doing the stimulation. It's systematic. We can stimulate after node within the same animal. We can do this at large scale and study both local and global networks. I really didn't get a chance to tell you about the feed forward versus feedback connectivity. So at the bottom, you'll see there are multiple pictures just saying that we can map thalamical-cortical connections, cortical-cortical connections, local cortical connections between different nearby regions, as well as I showed you, deep brain locations such as the amygdala. In addition, if you can image at a scale that allows you to differentiate superficial, middle, and deep layers of the cortex, you can distinguish feed forward versus feedback connectivity patterns. And that's something that we are working further on, looking at subcortical to cortical connectivity. I should also add that this method is pretty versatile in that we've used it on 2 different types of machines. We've used this in macaque monkeys, squirrel monkeys, as well as cats. And we've also tried this with and without contrast agent, and both of those approaches work. So it's not that difficult to do. I encourage you to try it. We have a paper that's under review that describes the procedure. So please let me know if you're interested. I'd be happy to get you started. Okay, I'm going to spend the last few minutes just talking about implications for AI. I really hope that because this can be done at large scale, I'm hoping that we can acquire lots and lots of information and see what the architecture of connectivity is. So I had shown you this picture before, showing you different types of local networks that we see. And this leads me to talk about potential motifs of networks. That is, I've summarized 3 that I commonly see. One is I refer to as lateral binding, where it's perhaps for binding different features, describing an object. So binding those features together and showing that those features are part of the same object. Another could be feed-forward transformation, whereby one have exemplars, such as chairs, individual chairs that converge into a more abstract concept of a chair. And this could be a circuit underlying categorization, where you have converging inputs onto a node. And then the feedback projections could be mediating context dependency, That is, this picture shows that the gray dot appears lighter than background or darker than background, even though the gray dot has equal luminance. This is a surround effect, and there are many surround effects in the brain, and feedback actions can mediate attention, gain control, and so this is a common kind of circuit that could be mediated by this third kind of motif. So these different motifs could underlie many types of computations in the brain. And if we can map them out and understand what is common from area to area, it would provide a more general understanding of brain architecture. So this image just summarizes how this mesoscale view changes our view of connectivity in the brain. So previous studies have shown that nodes are from area to area or sub-regions of areas to sub-regions of areas. Here we are showing that these are actually column-to-column connections. And so the connectivity patterns we're going to need to study are arrays to arrays, connectivity between arrays of columns. And so this changes sort of the mathematics of the description of the connectivity, and perhaps, it will impact how we view the architecture of the entire brain. So I'm hoping that those of you who are more mathematically or computationally oriented will get interested in this problem. And I summarize this problem here in this graphic, whereby the brain is represented by the sphere and areas by circles on the sphere. And then the orange and blue dots are columns within each area. And so the matrices represent the transformations between one area and another. The relationships, basically, they're representing relationships between arrays of columns. And so the mathematics would then need to describe how, if you have a pair of areas, how their relationships would then change if you add a third area and a fourth area and a fifth area and so forth. And so that can be a challenging problem, but hopefully somebody will tackle this problem. Okay, so we are, just to summarize and just mention a little bit about what I think the significances are. We're hoping eventually to get a Google map of brain connectivity at mesoscale. It does seem like a daunting task, but I believe that we can do it. Using the INS-fMRI method, it can be done systematically. And then the next step that we are hoping to embark on is to relate these networks to behavior and the dynamics of how these circuits are used in behavior. The issue of understanding brain architecture, I think, will have impact on other areas, such as brain-machine interface, where one could image the brain, understand more about its basic connectivity, and design brain-machine interfaces in a targeted fashion, where you know where the nodes are and you target those nodes for a specific purpose. So I envision such a brain-machine interface in visual cortex, for example. Targeting circuits for disease, where clinicians could really use this information to more specifically target nodes, such as for deep brain stimulation or other types of brain stimulation, and more specifically, and knowing what or being able to predict more what the effect would be of targeting different nodes. Instrumentation, I think MRI needs much more instrumentation. The multimodal MRI field is growing rapidly, and I think more funding needs to be devoted in this direction. And finally, connectional math theory. I really do think that this could be a breakthrough if somebody is really motivated to use this data to develop some kind of generalized understanding of brain architecture. And this would, in fact, not only affect our understanding of our own brains, but those that of other brains, as well as perhaps even of artificial brains. Okay, so this is my team. We are we work well independently on our own projects, as well as together on this very large scale project. And finally, I want to thank all the wonderful people who have contributed to this work, including my students and postdocs, other PIs at our institute at Zhejiang University. And if you ever want to look us up, we're at ZIINT, Zhejiang University Interdisciplinary Institute of Neuroscience and Technology, ZIINT. My collaborators in the US, Kathleen Gotard-Romansky, who brought tons of knowledge to the Amygdala project and really, really made everything so beautiful. Anatomists have very beautiful views of the world. And then Duco Jansen, who introduced me to the laser stimulation method. Originally, he began this in peripheral nerve, and then we were the first to take this to CNS. And my collaborators in the US, Rob Friedman, Nick Chernoff, and Jonathan Casey all contributed quite a lot to the INS projects. And I also thank my colleagues internationally who came to Zhejiang University to help us set up in our early days of 7T work, trying to figure out, well, how do we get bolt signal, you know? So there was quite a lot of fun there. So I always keep them in mind and thank them for all the contributions they make. And also I thank all my funding agencies for supporting this work. And I thank you all for your time and attention. Thank you.
Thank you, Anna. What a wonderful talk. If you can activate your camera and your mic, that'd be great. Thank you. Hi.
Hello, how are you?
Very good. Really enjoy your talk and your work in general. I'll be asking you the question from the floor, and I have also some question myself if we have the time. We have a first question about the potential clinical application of the INS method. That would be a little bit like deep brain stimulation, but you know, what is your perspective on that?
Well, I really am quite positive about the potential applications for clinic. So to give you, I talked about a little about deep brain stimulation. Give you another example, we've seen some very clear, organized and patchy connections between basal nucleus of the amygdala and the cingulate cortex, areas 23 and 24. And so, and these patches appear to fall in locations that underlie face and hand motor response during limbic response. And so it's potentially feasible to map out these limbic regions and really know what you're stimulating. And these could have applications for psychiatric disease, for example, and depression. Anterior cingulate is already being targeted as a potential treatment for depression. And one could further make this more specific and understand, well, what aspect of depression or what aspect of your motor behavior might be treated. So there are many potential applications, but one has to, of course, understand the circuits a little more. So I'm really hoping for a really neuroscience circuit-based approach to treatment of disease. I think it's possible.
So one example, if we take the anterior cingulate, will be maybe to stimulate an area that is not the anterior cingulate, but do project on this region and have maybe some less or some better consequences on the psychiatric behavior, reduction of depression, for example.
That's the idea. So
Yeah.
I think if we can understand the organization and topography of limbic behavior, that would be a huge help to treating these types of disease.
I think, you know, your work has been very inspirational for my work on the white matter, and yet, you know, I go to conferences and I find that the neuroscientific community is very corticocentric. To me, it's really a chance to speak with you, and if you could tell us a little bit more about the importance of white matter connection in the brain, just, you know.
Yes. So one of the goals, or one of the potential outcomes of this, is that we do understand the organization of white matter connectivity to a much higher degree. Diffusion tensor imaging has been used to study the white matter connectivity, but traditionally hasn't been done with sufficient resolution for us to see the specific organizations. And this, together with such structural approaches, may give us much more knowledge about how the white matter is organized. People have asked me, for example, well, why not stimulate the white matter? And that is a project I'm considering, you know, just to see how much organization is in the white matter. I don't think it's really known at this scale. So yes, I think that's a wonderful question, and indicates that there's another project we could do.
I think so neurosurgeons really use this very aggressive way, but yet like very successful way of stimulating the cortex and the white matter during neurosurgery to avoid removing part of the brain that are functionally relevant. And they had like a lot of effect when they were touching the white matter rather than the cortex. According to you, what would be really like, you know, what would be a functional unit in the brain? Can we really speak in areas, or should we really move on to large networks? Can I imagine like what would be your answer? But again, for the community of OHBM, I think it's important to insist.
Yeah, I
on
mean,
this point.
I truly do appreciate the challenges that neurosurgeons meet when they're in the operating room. You know, I have been collaborating with some neurosurgeons. Their aims can be quite different than for us neuroscientists, but I always try to remind people that while you're treating a specific location in the brain, that location is connected to a whole number of other nodes. And so it's not just that location that you're impacting. And while that may seem obvious, I don't think it's thought about enough in terms of treatment, because you need to treat the entire circuit in that sense. So knowing how this node that you're working on is impacting another node should be considered strongly as well. And that's not a critique, but it's a just,
of course, yeah.
I think it's a way forward. Yeah. I don't know if that answers your question.
It does. It does. It does. Another question is So I've always been shocked by work on the C2 architecture, for example, of Katrina Munz and Carl Ziles, showing how viable even primary visual areas can be. V1 can be very different from one person to another. And I was wondering, because you're building this matrix of connectivity based on different monkeys or cats, and I was wondering, what is the impact of variability, inter-individual variability on the reliability of this matrix? How do you handle this level of complexity, which is like every living species is not exactly the same inside the same species and between species, of course?
Right. Excellent question. Of course, I think much of MRI has been based on the average of multiple subjects, and this has taught us an enormous amount. And in our work, we also plan to do some averaging. You know, typically with monkey work, you can't do too many monkeys, but you know, maybe 2 or 3 or 4 monkeys in a single study that is feasible. But I think what we contribute more to that is not as well understood is the relationships of different networks within a single individual. And I do expect, number one, of organized pattern, number one, between individuals for example, for treating individuals. So for example, one could image at high resolution an individual, and then based on that individual's scans to really target specific locations. And that speaks very strongly to a future approach to personalized medicine. I really do think that this is going to become feasible. I mean, I don't mean doing a connectome in every individual, but at least finding out enough information such that one can predict from these generalized maps, one can predict what that local organization is going to be and increase the understanding of how to treat that local region.
Okay, so what would be your prediction? Would you think like inter-individual variability will be like a random noise that will be distributed, or will it be like cluster of people that have different configurations? So they're like you have a group of people that are kind of with this kind of anatomy, and other people that are. Do you see what I mean?
Yeah. So, well, that would be a very interesting fallout if there were clusters of types of anatomies. That would be very interesting. But that's I would say a more future point. Initially, it would be simply understanding the rules of how the anatomy can be organized. I mean, what are the possibilities? And if we see different examples, different exemplars of certain types of layouts, we can start to understand, well, what are the rules? So for example, we know some of the rules underlying how maps are created in the brain, multimodal maps. And if we can generate such rules for connectivity as well, interareal connectivity, that would be a huge breakthrough. So, for example, you know, Hebb's rule has dominated our understanding of how multiple maps are interdigitated within a single area, right? But we don't really have an understanding of how that in turn predicts the connection patterns to another area. And this is something that I really think can be addressed by modeling methods. And that would be so instructive. With a little bit of data that we can provide, providing some constraints, I think there could be a lot of work, modeling work, that would be very instructive to point out what are some of the potential rules for setting up these maps.
Thank you very much. I have like some signs on the chat that we have to stop unfortunately, it's very interesting exchange and discussion on your research. I hope we'll
Well,
have the
thank you so
chance
much
to
for the
meet
questions.
again. And
Yeah, I would love to discuss more.
all right. Well, with this, so thank you so much for your talk and for presenting to OHBM. Thank you so much for your research and your patience answering the question. And well, I'm looking forward to meeting you again. Thank you. Bye-bye.
Bye-bye.